This module is called Annotating Images. In the previous modules, we've covered how images are acquired, represented, visualized, and now stored. Now we'll begin interacting directly with images in QuPath. Annotation is one of the most fundamental tasks in digital pathology and image analysis. Before we can measure tissue regions, detect cells, classify structures, or train machine learning algorithms, we often need to define specific regions of interest within an image. In this module, we'll explore the annotation tools available in QuPath, discuss the differences between annotations and detections, and learn how annotations can be used to generate quantitative measurements. In this module, we'll focus on three key questions. First, what are annotations and why are they important? Second, how are annotations different from detections in QuPath? And third, how can we use annotations to generate measurements and quantitative data? By the end of this module, you should be comfortable creating annotations, understand when to use annotations versus detections, and understand and know how to access measurements derived from selected regions of interest. Drawing annotations is a routine task in QuPath and serves several important purposes. One common use is making measurements. For example, we might draw around a tissue region to calculate its area, or use a line annotation to measure distances. Annotations are also used to define regions where downstream analyses should occur. For instance, we might restrict cell detection or segmentation to a tumor region while excluding surrounding tissue. A third application is creating training data for machine learning or classification workflows. By manually identifying representative regions, we can teach algorithms what different tissue types or structures to look for. You may encounter similar concepts in other software packages under names such as regions of interest or ROIs, overlays, or segmentation regions. Although the terminology varies, the underlying idea is usually the same, defining specific areas within an image for visualization, measurement, or analysis. An important concept in QuPath is the distinction between annotations and detections. Annotations are flexible objects. They can be edited after creation, reshaped, expanded, reduced, or otherwise modified as needed. Because of this flexibility, annotations are relatively memory intensive. Every time the image display updates, QuPath needs to redraw these objects. For that reason, performance may slow when working with a very large number of annotations. Fortunately, most projects only require hundreds or perhaps thousands of annotations, and so most computers can handle this. Detections are different. Detections are designed for efficiency. Once created, their regions of interest are generally fixed and cannot be modified in the same way as annotations. Because they are lightweight and optimized for performance, QuPath can efficiently manage millions of detections provided the objects remain relatively small, such as individual nuclei or cells. In practice, annotations are usually used to define regions for analysis, or detections represent individual objects identified within those regions. In this exercise, we'll become familiar with the annotation tools available in QuPath. Open the QuPath documentation and navigate to the section on Annotating Images. Experiment with each annotation tool and observe how they behave. As you work through the tools, consider the following questions. What are the similarities and differences between them? Which tools are best suited to tissue regions, irregular structures, or precise measurements? How easy is it to modify an annotation after it has been created? And what practical tips have you discovered for switching between tools efficiently? The goal here is less about producing perfect annotations and more about becoming comfortable with the workflow. I'll pause here to give you a chance to do this exercise yourself. I'll now demonstrate how to perform the steps of this exercise. We'll start by going to the documentation for QuPath. Here I'm on the landing page for the documentation of QuPath. If I go to Getting Started and Annotating Images, we'll then get to this page where you'll recognize the image from my presentation slides and information about each of the icons that are in the toolbar. Returning to our project, we can start using these tools, such as drawing squares, drawing circles, lines, and other such tools such as polygons, where we can either click and double click to finish. I'll demonstrate this again, showing the inputs in the bottom left corner. So here I'm clicking for single points of the polygon, and then when I'm ready to finish, I double click. A similar thing is available for open polygon, but for the closed polygon we can also click and drag for more complex geometries. We have the brush tool, which as you zoom in or out of our image, we can see that the brush tool adapts the size that you're brushing. We also have, if we reset to the whole image, we can also use the one tool. Now this considers a radius around the point you're clicking and selects pixels that are similar to where my cursor is currently. Therefore you can get nice complex geometries which follow the pixel intensities. Now be aware this changes depending on the channels that are in view. So if we now turn off our red and green channels and only leave the nuclei in our image, we can see that the radius and what we're following is adapted to the nuclei. And finally we have our points annotation, where we can draw individual points, adjusting how big we want them to show, and have multiple different sets of points with maybe different classifications. Now all of these annotations under the analysis appear under the analysis pane under an annotation tab, and here you can see the list of annotations that we've drawn. You can also see some classes that we can assign them as. So for example, these points here I can say refer to the immune cells, and that this set of points here can maybe refer to necrotic cells. Also we can have regions assigned as tumor and stroma, and we can also do that by selecting them in the annotation list and setting the selected. You can also see there's some information below taking measurements of our annotations. Now something that I'd like to point out are there are some shortcuts with our mouse. For example, if we press on the middle mouse button, it will bring up whatever annotation we last did. So the last annotation I did here were points, so the points annotation window has appeared. If, say, I use the brush annotation and then click off and press the middle mouse button, I can then go back to the brush. I can also then toggle off the brush annotation if I want by selecting the mouse button to go back to panning. If you found that you weren't able to draw annotations, maybe this is because the selection mode had been toggled on. We now see that each of our icons have become dashed, and it's with this that we don't create annotations, but we can select annotations. So for example, with the polygon tool, I can lasso around annotations, and those annotations that are within can then be selected. We can also toggle on and off the annotations that are in view. So if we select this icon or press A on our keyboard, we can see that only the selected annotations are revealed, and as soon as they're not selected, they also disappear. We can also provide shadings to our annotations to help maybe understand whether we're within an annotation area of a certain class. You'll also notice that annotations can overlap one another. However, you might find it difficult that with some annotations having to merge with others. For example, here we have an annotation and I have the brush tool on. If I try and draw a new annotation over it, I can't. I can only add to what is already there. However, if I switch to the polygon tool, I can draw a new annotation overlapping the annotation already there. Now that we've created several annotations, let's consider the situation where we may want to save those annotations and share them with others, or we want to save a version of the annotations so that if we do edit our annotations further and think they're incorrect, we can always revert back to an earlier version. So to do this, we're going to follow these steps of the exercise. We're going to save our annotations by selecting File, Export Objects as a GeoJSON, and we'll keep the default settings and select OK. We'll save the GeoJSON file somewhere reasonable, in this case the QuPath project directory, and then back in our QuPath project we'll make a duplicate of our image, removing all annotations already there. We'll then open that image in the viewer. We'll then allocate our GeoJSON file in the directory of our QuPath project, and then simply select and drag it into the QuPath viewer to load it back in. I'll pause here to give you a moment to perform this exercise yourself. I'll now demonstrate how to perform the steps of this exercise. Back in our QuPath project, we have our annotations. We want to make sure we save the annotations to this image by clicking Save. But to then export the objects as a GeoJSON, we can go to File and Export Objects as GeoJSON. We'll keep the default settings and click OK, and then save the annotations in our QuPath project. We'll now go back into the Project tab in the Analysis pane, and we'll right-click on the fluorescent cells image and duplicate image. Here we want to give our duplicate file name. Here we'll just keep it as the default, but we won't duplicate the data files. This means any of the analysis that we have done on top of this image. So when we then switch towards it, we'll also be prompted to save changes to our image currently open in the viewer. We'll click yes, even though we have saved, and see that we have our fluorescent cells image open with none of the annotations. So to open the project directory, we can simply right-click in the image list, go to Open Directory for Project. We can see that our GeoJSON file is in our QuPath project, and so we can select and drag onto the QuPath viewer. Now here we can see that only one of the annotations has been saved. This is because you may have noticed that when I was back in the original image, I had only this annotation selected. To make sure that all annotations are saved, we can go through the same steps again, being careful that whether we want to overwrite the file already there. In this case, we're happy to do that. You can see QuPath now says that we saved 19 objects because we have 19 annotations. we'll save the changes there and simply drag and drop the file. Now, because we already have an annotation, we'll have this prompt which says you may want to update the IDs for the annotations. This is strongly recommended. We will do this in this case. But the reason why it has done this is that when you open an annotation, we see that we have one annotation here from before, and so there is a duplication of the same annotation. So be aware that when you're opening annotation files, it will add on the annotations to what might already be in the image. Once the annotations are loaded, we can also save these annotations to the image. Because, for example, if we did not, and we didn't save that progress, and said no to the prompt of whether you want to save it, and then revisit the image, we'll see that all the other annotations are not there anymore, and only the one that we loaded in before. This is why it's important to keep saving your work throughout your analyses. We'll now consider how we can take these annotations and measure these regions of interest. To do this, we'll open the documentation and look at the two options available to us. We'll follow the instructions on how to access the measurements in a measurement table within QuPath, but also look at how we can export the measurements into a file which can be used in other programs. I'll pause here to give you the opportunity to perform this exercise yourself. I'll now demonstrate how you can perform the steps of this exercise in QuPath. In QuPath, we'll go back to the image which has multiple annotations. Here, we can view measurements for our annotations by going to the Table tab and Show Annotation Measurements. Here we can see a table appears in a new window, where there is a thumbnail for the annotation, which image it relates to, the type of object it is, and some measurements. We can also interact with the table dynamically. Because our viewer is currently zoomed out onto the whole image, we can also zoom in and double click on entries so that it will take us to those annotations. We can also interact with these annotations by ordering them. For example, say if we're interested in the area, which has the largest or smallest. Here we have some points, so they're not going to have any area, but then we have some other annotations which have small or large areas. We can also filter columns in the bottom. So for example, if we're interested in where the centroid of these annotations are, we can see that we've now filtered our columns to only those that contain the keywords. There's also the ability to plot this data, where we can select which measurement we would like to plot. When we're finished interacting with the data and want to preserve the measurements for downstream analysis, we have two options. We can simply save this table by either File and Save or the Save icon here, or we can exit the measurements table and go to Measure, Export Measurements. Here we have the ability to export measurements from across multiple images in our project. We can then simply select the images that we would like to export in our measurement table. We can then choose what our table should be called and whether it should be tab separated or can be a compressed version. We can also change this to a comma separated table or a semicolon separated table. Now here there's the option of what is it that we'd like to summarize in the table. In our case, we simply want to summarize by annotations. And there are some other options which we can read about in the documentation. Whereas here, we'll simply select Export. Now we have a table saved in our QuPath project called Measurements. And this is a tab-separated variable. Depending on your operating software and programs you have already installed, we can open the file with Notepad or a text editor and see that each column is separated by a tab and each row is an annotation from our measurements. You can also set it up so that a program such as Excel can automatically recognize tab-separated variable files and then interact with the data in a spreadsheet. Let's revisit our objectives. We've learned what annotations are and explored the important roles they play in image analysis workflows. We've compared annotations and detections, highlighting their different purposes and performance characteristics. We've seen how annotations can be converted into quantitative measurements that can be viewed, exported, and analyzed. These concepts provide the foundation for many of the more advanced workflows we'll encounter later. To conclude, annotations are one of the most important tools in QuPath because they allow us to define regions of interest within an image. These regions can be used for measurements limiting analyses to specific tissue compartments, or generating training data for classification workflows. Remember that annotations are editable and flexible, making them ideal for manually defining biological structures. However, this flexibility comes at the cost of increased memory usage. Detections, on the other hand, are lightweight objects optimized for large-scale image analysis. They are typically used to represent cells, nuclei, or other automatically identified structures, and are common outputs of cell segmentation algorithms. A useful way to think about the relationship between the two is that annotations define where analysis should occur, while detections describe what has been found within those regions. Finally, annotations are not just visual objects. They can generate quantitative measurements that can be inspected within QuPath and exported for further statistical analysis, making them a critical bridge between image viewing and data analysis. Thank you for joining us for this module, and I hope you can join us for the next one, the module on quantitative image inspection.